Here we'll look at the bacteria Bdellovibrionata, which was previously in the class Delta Proteobacteria. However, recently a proposal to reclassify these classes into different phyla was published and accepted. So now, after this paper, Wade et al. 2020, Bdellovibrionata has its own phylum, Bdellovibrionata, class Bdellovibrionia, order Bdellovibrionales, family Bdellovibrionaceae, and genus Bdellovibrio. So you can find the latest list of prokaryotic names with standing in nomenclature, the one that is formally recognized at this website. Species is Bdellovibrionata. It was first described in 1963. It's a gram-negative obligate aerobic bacteria, and as of now there are only 4 species: Bdellovibrionata, Bdellovibrioexovorus, Starri, and Stolfi. Okay, these bacteria are found in river water and soil. Interesting fact about this bacteria is it parasitizes other gram-negative bacteria by entering their periplasm and feeding off their host. So this is one of the rare bacteria that actually predates on other bacteria. So there are it has two lifestyles. One is the highly motile attack phase and the slow-growing Bdelloplast phase. Enters the host cell, it will start consuming the cytoplasm of the host cell and elongating. Okay, it loses its motility, it loses its flagella, and becomes a very, very large cell, a Bdelloplast. However, once the food supply is exhausted, it will begin to septate itself, form septates, and then be released from the host and to enter the motile phase again. In the motile attack phase, it swims very fast, about 160 micron per second. So this is about 100 body lengths in a second. So that is relatively very fast. So it will swim at this rate and then collide with its prey, will attach and enter through a hole in the periplasm. Then it will use hydrolytic enzymes to break down the host cell to elongate. Okay, so as mentioned, once the host cell nutrients are exhausted, septates will be formed for the progeny Bdellovibrios. So for one cycle, which takes about 1 to 3 hours, it can produce from 3 to 6 progeny from a single E. coli cell. Showing the different phases, the 2 different phases in Bdellovibrio life cycle. So this is the free living attack phase. So once it enters, it attaches. So here you can see the time it takes for each phase to occur. And 4 hours, the Bdelloplast lysis occurs and the release of the progeny Bdellovibrio. Here we have the host dependent lifestyle, and here are the host independent growth. So host independent growth will be in biofilms or in liquid. So usually you grow them in co-culture with E. coli in an overlay nutrient agar at 30 degrees Celsius for about 1 week. So this bacteria has a biotechnological potential where there is interest in the application of Bdellovibrio in eradicating pathogens, for example, in eradicating Salmonella enteritidis in eggs. Here we'll look at the phyla Cyanobacteria Chloroflexi and Chlorobium that contains photosynthetic microorganisms. So Cyanobacteria carries out oxygenic photosynthesis and nitrogen fixation. So these are very important components in the sea, in the aquatic habitats, where they are able to fix nitrogen and also carry out primary production in the aquatic habitats. So role in the food webs of aquatic habitats. They can be unicellular or filamentous, previously known as blue-green algae, and examples of cyanobacteria are Shinnecococcus, Ionobina, and Trichodesmium. However, for Chloroflexi and Chlorobium, both of them carry out an oxygenic photosynthesis, which means their photosynthesis does not release oxygen. And from here you can see the difference between Chloroflexi and Chlorobium, where Chlorobium requires hydrogen sulfide for CO2 reduction, whereas for Chloroflexi they take up organic compounds for CO2 reduction. Names for Chloroflexi is a green non-sulfur bacteria, whereas for Chlorobium they are green sulfur bacteria. So examples for this phyla is Chloroflexus, and for this is Chlorobium.